In 1866, DNA first started with the concept of genetics. Gregor Mendel, an Augustinian monk, discovered the basic principle of genetics. Mendel conducted experiments on pea plants attempting to crossbreed. He identified the 7 characteristics: plant height, pod shape and color, seed shape and color, and flower position and color.
In 1869, Swiss physiological chemist Friedrich Mischer first discovered and coined the term nuclein, which is found in the nuclei of a human white blood cell. This nuclein is what we call DNA, or deoxyribonucleic nucleic acid in the present.
The genic movement evolved. This is a dark chapter in the history of DNA. Further research about the genetics by Mendel was done during this time, and this resulted to the ability to predict traits and characteristics of a human being. The researchers wanted to control the breeding of human beings so that only the people with the best genes could reproduce and improve the species. This resulted to discrimination and racial hatred.
In 1902, Sir Archibald Edward Garrod is the first to associate Mendel's theories with a human disease. Also the first time that a genetic disorder had been linked to inborn errors of metabolism, which he referred to his belief that certain diseases were the results of error or missing steps in the body's chemical pathways.
By the 1940s, genes were known to be the discrete units of heredity, as well as the generating enzymes which controlled metabolic functions. However, it wasn't until 1944 that DNA was identified as the transforming principle by Oswald Avery. Avery, MacLeod, and McCarthy combined to determine which substance was responsible for the transformation when pneumococcus was mixed with harmless bacteria. Avery noted that the substance did not seem to be protein or carbohydrate, but rather a nucleic acid, and was revealed to be DNA.
In 1944, scientist Erwin Chargaff had read Oswald Avery's scientific paper, which identified DNA as the substance responsible for heredity. In 1950, he summarized his 2 major findings regarding the chemistry of nucleic acids. First, that in any double-stranded DNA, the number of guanine units is equal to the number of cytosine units, and the number of adenine units is equal to the number of thymine units. And second, that the composition of DNA varies between species. These discoveries are now known as the Chargaff's rule.
Rosalind Franklin photographed crystallized DNA fibers using X-ray crystallography.
In 1951, James Watson visited the Cambridge University and happened to meet Francis Crick, and they remained at the university to study the structure of DNA at Cavendish Laboratory. Using available X-ray data from Rosalind Franklin, they were able to solve the puzzle that had baffled scientists for decades. And in 1962, they were awarded the Nobel Prize for Physiology or Medicine along with Maurice Wilkins. However, Rosalind Franklin was not credited for the discovery of the structure of the DNA.